Hey everyone, so today our group is going to give an overview of the neuroscience research methods. And we'll start off by talking about behavioral and lesion studies, which are two ways of studying the brain that fall under the branch of behavioral neuroscience. Behavioral neuroscience examines the structure-function relationships underlying the behavior of animals and humans. And lesion studies are typically a type of experimental approach that can be used to study the brain, but there are occasional exceptions when lesion studies are not experimental, like the case study of Phineas Gage. In experimental studies, like lesion studies, any manipulations of the brain can be considered independent variables, and the effects that these manipulations of the brain have on behavior are the dependent variables. And by generating lesions surgically, chemically, or electrically, and knocking out a specific part of the brain, lesion studies can help us to infer what the functions of these specific brain regions are by examining the effect of the lesion or knockout on the behavior of the individual. And as you can see in this GIF in the top right corner, lesions are invasive methods that directly go into the brain. And lesion studies are typically performed in animals rather than on humans for several reasons, including ethical reasons. However, we can still use the results and information we obtain through animal brain lesion studies and apply it to human brain functions as well.
Another popular method for studying the brain is through drug studies. This involves the researcher using drugs to directly alter brain function but not structure of its patients. This allows the researcher to evaluate how the brain responds to specific drugs, as well as target different parts of the brain in the study. Drug studies can be conducted either within or between subjects. Conducting a within-subject study means the same group of subjects will be treated as the control and experimental groups. Within-subject studies are best for small sample groups and eliminate the chance of variability due to differences between individual subjects. Alternatively, between-subject studies use different groups of subjects as control and experimental groups. This type of study requires a larger sample group, but also allows for more time efficiency as the placebo and experimental groups can be evaluated at the same time, and allows for larger generalizations to be made from the data. One example of a drug study is from an article about the introduction of MDMA into rats leading to neuronal death. The image to the right shows the difference in neuronal presence prior to MDMA introduction and after MDMA introduction. This is also a within-subject study because it is the same rat's neurons being evaluated in these two images. The following method of brain studies, which will be the main focus of the remainder of this presentation, is brain imaging. The specific methods we will discuss either directly or indirectly measure brain activity or create images of the brain, all through non-invasive practices. Normally, these methods are used to evaluate which parts of the brain are impacted or altered from illnesses or neurological disorders. This is because these imaging methods allow neurologists to see exactly which pathways or structures of the brain are experiencing abnormalities. The four main types of brain imaging that we will be discussing for the remainder of this presentation are fMRI, PET scans, CT scans, and EEGs.
So the first type of brain imaging method we're going to discuss today is the electroencephalogram, aka the EEG. So this directly measures electrical activity within the brain through the use of electrodes that are placed on the scalp, as seen here. So this gives you a summation of all electrical activity in the brain, but it cannot give you information about individual brain regions. So the output of an EEG looks somewhat like this. It's kind of like a brain wave. And this kind of method is commonly used in sleep studies. So next we have the PET scan, as it's commonly known. This uses radioactive tracers injected into the brain to visualize brain activity. So a commonly used tracer is glucose, and the output that you get is these kind of very colorful images of the brain. That gives you a little bit more information about the area in which the activation is occurring. However, this method has become more outdated with the use of fMRI and MRI these days.
This method we'll be discussing are fMRIs. Functional magnetic resonance imaging is a type of neuroimaging method used to indirectly measure brain activity. Exposing the brain to multiple magnetic fields allow fMRI to detect changes in blood flow and oxygenation that occur in response to neural activity through blood oxygen level dependent contrast, also known as BOLD. Areas of the brain that are more active tend to receive higher levels of oxygenated blood and then color-coded in images to represent the specific areas of the brain activity. You can see the color-coded regions in this figure here, representing active parts of the brain once neural stimulation occurs. fMRI's popularity is derived from its widespread availability, non-invasive nature, relatively low cost, and good spatial resolution. It is being increasingly used as a biomarker for disease, monitor therapy, as well as to monitor growth of brain tumors and determine how well the brain functions after a stroke or even Alzheimer's diagnoses. And finally, CT scans combine a series of X-ray images taken from different angles around your body and computer processing to create cross-sectional images of bones, soft tissue, or organs inside your body. It provides greater detailed information compared to the conventional X-ray image. Regular X-rays use a fixed tube to send rays in only one direction, while CT scans use a motorized X-ray source to shoot narrow beams as it rotates around the patient, as you can see in this figure here. The image slices created can be displayed in a 2D form or stacked together to create a 3D image, which can reveal abnormal structures, internal injuries, or can even be compared over time to assess the progress of tumor degradation through radiation therapy.